In some cases, it may be necessary to merge samples that have been acquired either on separate or same lanes. This is because not sufficient number of cells have been collected for a sample on a single lane. Assuming the experimental conditions are sufficiently similar, one can merge cells from one sample with another. MAS will conduct this exercise in a fully traceable manner, that is, it is possible to unmerge a merged sample. To merge samples, simply click on the two samples or more that you want to merge and click on merge. This is also the screen where you can change the names of the samples and also add annotations to samples. You can change the name by clicking on the edit icon and giving it a name that is more appropriate. Finally, to add annotations, click the download template to get an Excel file which you can add additional columns of data as annotations. Here you can add additional annotations like treatment conditions or any other experimental variables that you might want to record against each sample. Once the Excel file has been edited, click on Select Annotation File to bring that Excel file back into MAS. And then click Upload. You should now show the new annotations that have been added to each of the sample, and these annotations will be available later downstream for analysis. Once the samples have been annotated, the next step is to define cell types in MAS.